everyone. In this video, we are going to discuss about the development of male gametophyte in angiosperms. In previous video, we have discussed about the process of microsporogenesis, and there we have observed that there are pollen mother cells or microspore mother cells present inside the microsporangia of all anthers. Okay, so this pollen mother cell undergoes mitotic cell division. Okay, so you can see here this is pollen mother cell. Another name for this pollen mother cell is microspore mother cell, and this is generally diploid. Okay, and this pollen mother cell undergoes meiotic cell divisions, that is meiosis 1 and meiosis 2. This is reductional division. That means the type of cell division in which the chromosome numbers in daughter cells are reduced to half. Okay, and after meiotic cell division, or after undergoing meiotic cell divisions, this pollen mother cells forms 4 haploid microspores. Okay, 4 haploid microspores. Now in initial stages, these 4 haploid microspores are actually held together with each other. Okay, now there is a particular polysaccharide which is called callose. So this polysaccharide actually holds or binds all these 4 haploid microspores in the form of tetrad inside microsporangia. Okay, if you remember when we were discussing about the transverse section of anther, this is microsporangia, and outside microsporangia the next layer of cells is known as tapetum. Okay, so these are the cells of tapetum. Okay, so these cells of tapetum, they secrete an enzyme which is called callase. Okay, the name of this enzyme is callase. So when microspore tetrads are present inside, okay, or when microspore tetrads are formed inside this microsporangia, this callase enzyme will digest the callose, which is actually holding all these microspores together in the form of tetrad. And after digestion, what will happen? All these microspores now are free in the microsporangia. Okay, they are present as a single individual inside the microsporangia of anther. Okay, so this one is now your pollen grain, or it can also be called microspores. Okay, but right now this pollen grain or microspore is not ready to be released from the anther for the process of pollination. Before releasing out from the anther for the process of pollination, this pollen grain or microspore must undergo certain changes. Okay, and after it undergoes certain changes, it then develops into a male gametophyte. Okay, so now we'll begin and we will understand how this pollen grain changes into male gametophyte. Initially, this pollen grain is present like a simple cell. Okay, it has a plasma membrane, then it has a haploid nucleus. Okay, and then it has cytoplasm. Okay, plasma membrane, haploid nucleus, and cytoplasm. Okay, now first what happens is this pollen grain, while it is undergoing some changes to develop into male gametophyte, this pollen grain first develops 2 layers around itself on the outer side. Okay, it will develop 2 layers around itself. Now the inner layer is actually continuous layer, continuous and smooth layer. Okay, and this inner layer is known as intine. Okay, what it is called? Intine. Okay, and the outer layer around pollen grain is actually having a spiky structure, or you can say a pointed structure, and this layer is not continuous. There are certain gaps in this layer. Okay, in between this layer. Okay, this outer layer is known as exine. Okay, now this intine and exine actually helps in a kind of protection, or they protect this pollen grain from the unfavorable conditions. Okay, and after the production of this intine and exine, Now this pollen grain which is present inside these two protective layers, this pollen grain undergoes mitotic cell division. Okay, so this pollen grain now undergoes mitosis and produces two uneven cells. Okay, uneven cell because the size of the cell is different. Okay, so maybe this is intine and outside we have exine. Okay, okay, this is intine and exine. Now inside we have two uneven cell. One is very large cell. Okay, this particular cell is actually known as vegetative cell, and or the another name is tube cell. Okay, and it has its own haploid nucleus, and then we also have inside this another smaller spindle shaped cell. Okay, this is a spindle shaped small cell, and this cell is known as generative cell. Okay, generative cell. Okay, now this larger cell is known as vegetative cell or tube cell because this larger cell is not directly involved in the process of reproduction. Okay, this larger cell is actually having a very dense cytoplasm. Okay, it is having a very dense cytoplasm because there is, you know, large amount of food reserve present in this larger vegetative cell. Okay, and this cell is also called tube cell because we have studied in lower grades that whenever pollen grain falls on stigma, so this pollen grain also produces a pollen tube. Okay, so this tube cell or vegetative cell gives rise to the pollen tube of germinating pollen grain. Okay, whenever it falls on the stigma. The second type of cell, the smaller, the small and spindle shaped cell is known as generative cell. Okay, it is called generative cell because the cell produces male gametes. Okay, the cell will produce male gamete. You can see here in this picture, this thick outer sculptured wall is present. Okay, and in between it is having a pit. This gap which is present between these exine layer, okay, this gap is actually known as germ pore, and this gap is present here. This is important because pollen tube, when pollen tube is produced by vegetative cell, so this pollen tube actually comes out of pollen grain through this germ pore. Okay, this pollen tube can come out of pollen grain through this germ pore. Now, why it is important? What is the importance of germ pore? Why this pollen tube cannot come out of exine? This is because we have studied in the previous video that cells of tapetum, they secretes Ubish bodies. Okay, and these Ubish bodies are actually coated with a polymer, a biopolymer, and the name of this biopolymer is sporopollenin. Okay, sporopollenin, and sporopollenin is the highly or highest resistant substance known in the living world. Okay, it is highly resistant to the temperature, it is highly resistant to the pH, that means to the acidic condition as well as to the alkaline condition. For example, if an animal like human being, us, if we consume pollen grains, okay, so nothing will happen to this pollen grain. Okay, it will just come out of our body through other fecus, and after coming out of the body in fecus, even after that, if it falls on the stigma of any flower of its own species, this pollen grain has the ability to develop or has the ability to, like, fuse with female gamete. Okay, so as this sporopollenin is present in this exine, this is the highest resistant substance ever known in the living world. So this pollen tube cannot break the exine and come out of it. Okay, that's the reason the germ pores are present in the exine. Okay, and hence exine is not continuous layer. Okay, it is present in a discontinuous manner. This exine is made up of sporopollenin, and this intine is made up of cellulose and pectin. Okay, next is like initially when this generative cell is present, when this generative cell is formed. Okay, so this generative cell is actually covered with a callose layer. Okay, and this callose layer is actually, separating the cell from the pollen grain, and this callose dissolves later on, and then this generative cell becomes naked, and it is present in spindle shaped form, and it simply floats here and there in the vegetative cell. Okay, and this generative cell has transparent cytoplasm. This generative cell has transparent cytoplasm. Now, in most of the angiosperms, what happens is, almost in more than 60% of angiosperms, this pollen grain is released from the anther during this two-cell stage. Two-cell stage means intine is present. Okay, this blue color layer shows intine. Exine is present. Okay, we have this germ pore. Okay, and inside this vegetative cell is there, or tube cell that is having its own nucleolus, nucleus and cytoplasm. And then we also have generative cell. This generative cell is also having nucleolus, nucleus and cytoplasm. So now two cells are present inside pollen grain, vegetative cell and generative cell. Okay, so in most of the angiosperms, these pollen grains are released when they are present at a two cell stage. Okay, but in some of the angiosperms, what happens is, before releasing outside the microsporangia, or before releasing outside the anther, these this generative cell present inside the pollen grain undergoes cell division, and it produces two non-motile male gamete. Non-motile means something which cannot move. Okay, so this generative cell divides twice and produces two non-motile male gametes. Now, how many cells are present now? One tube cell or vegetative cell and two uh, male gamete cells. Okay, non-motile male gametes. Okay, so totally there are three cells. So in some angiospermic plants, the pollen grains are released at three cell stage, and those pollen grains which are released at two cell stage, when they fall on the stigma and when they start to germinate. Okay, so at that time this generative cell undergoes cell division and produces two male nuclei. So there are certain facts about pollen grains. Okay, pollen grains of many species, they can cause chronic respiratory disorders in human beings like asthma and bronchitis. Okay, this is because some people are actually allergic to pollen grains, and if they inhale these pollen grains by mistake along with the air, so these pollen grains causes bronchitis and asthma in them. Okay, one of the example for such type of plant is Parthenium hysterophorus. Okay, so Parthenium hysterophorus. It is written in italics because all the botanical name of plants should be written in italics. This is the rule. Okay, so this plant is a Parthenium plant, a most commonly available. Everybody must have seen this plant in your real life. The origin of this plant is actually not from India. India has actually imported wheat from the foreign countries at that time. Along with the imported wheat, this Parthenium species has come to India. Okay, the Parthenium species has come to India along with the imported wheat. Okay, and this is a very common weed. Okay, this is actually a very common weed. So it's of no importance, no importance to any human beings or animals. Animals might be grazing them, but obviously those who are allergic to this pollen grains, they do not. Okay, the next fact is that these pollen grains, they are highly rich in nutrients. Hence, pollen tablets and pollen syrups are available, especially in foreign countries. Okay, so people in foreign countries, they consume these pollen tablets and pollen syrup as a food supplement because these pollen grains are highly rich in nutrients. Okay, and one more point to be noted here is that these pollen consumptions can increase the performance of athletes and race horses. Okay, so these pollen consumptions can increase the performance of athletes and race horses. So that's the reason they are consumed as food supplement in most of the countries. The last point about pollen grain is that the viability of these pollen grains varies from species to species. Okay, the meaning of term viability is that after releasing from anther of the plant, for how many days these pollen grain has the ability to germinate if they fall on the stigma of the plant. So how long they have the ability to germinate, okay? And this viability of pollen grain varies from species to species. Some plants like rice and wheat, they have this viability of only for 30 minutes, okay? So this pollen grain of rice, wheat, then other millets like bajra and then ragi and all. So the pollen grain of these cereals, they are viable only for 30 minutes. But there are certain plants like rose, potato, and other leguminous plant like olive pulses plant, soybean, groundnuts, and all. Their pollens they remain viable for 30 days. Okay, so almost for 30 days. But if we want, we can preserve these pollen grains for even longer period of time. Okay, and for preserving pollen grain, we require liquid nitrogen. Okay, so nitrogen gas has to be compressed, okay, and then it has to be changed to liquid nitrogen. Now obviously these gas molecules, they have higher kinetic energy, so in order to reduce their kinetic energy, we have to decrease the temperature. So liquid nitrogen, or you can say nitrogen gas, or the nitrogen element, remains in the form of liquid at 196 degrees Celsius, okay, at minus 196 degree Celsius, okay. So pollen grains can be stored in liquid nitrogen at minus 196 degrees Celsius for many years or for longer period of time, so that they can be used as pollen banks, okay. Like you must have heard about in vitro fertilization, that is IVF. We will learn about this in the next chapter. Okay, so for IVF, the sperms from the male and ovum or egg from the female are taken out, and then they are preserved again in the colder conditions, okay, for artificial insemination, okay. So similarly, these pollen grains are also preserved for years in liquid nitrogen at minus 196 degree Celsius temperature, and hence pollen banks are available, okay. So these were the facts about pollen grains, and I hope you people are clear with the process of microsporogenesis and development of male gametophyte. If you have any query about this, please mention your questions in the comment section below, and I will definitely revert back with the correct answer. Take care till then. Bye bye.